Hello, today we will be talking about the Duffy blood group system. The Duffy blood group system was discovered in 1950 when Cutbush and associates identified anti-Duffy A in Mr. Duffy. A year later, in 1951, Eakin and associates identified anti-Duffy B in a woman who had multiple pregnancies but had never been transfused before. In 1955, Sanger and associates reported the Duffy null phenotype Duffy A and Duffy B negative, and in 1975, Miller and associates reported that the Duffy A, Duffy B negative phenotype was resistant to infection from Plasmodium vivax and Plasmodium knowlesis, which are known to cause malaria. The ISBT symbol for the Duffy blood group is FY, and 008 is the numerical designation. The Duffy blood group is composed of 5 antigens: Duffy A, Duffy B, Duffy 3, Duffy 5, and Duffy 6. Previously, there were 6 antigens, but Duffy 4 is no longer recognized by the ISBT. The Duffy gene is located on chromosome 1 and encodes for the glycoprotein called the Duffy antigen receptor for chemokines. Duffy A and Duffy B alleles are inherited as Mendelian codominant and are known to be polymorphic. The Duffy null phenotype, Duffy A and Duffy B negative, is a result from being homozygous for either the GATA1 mutation or the silent allele FY. The GATA1 mutation can only be found in Africans and African Americans, and is identical to the Duffy B allele. It is the result of a single nucleotide polymorphism in the GATA promoter region of the Duffy B allele. This causes the binding site for the red cell-specific GATA1 transcription factor to be disrupted, preventing expression of the gene on the red blood cells. As a result, no Duffy glycoprotein or Duffy B antigen will be found on the red blood cells, but they will still be present on other tissues in the body. Homozygous individuals will phenotype as Duffy A and Duffy B negative, but they cannot make anti-Duffy B because the antigen can still be found on other tissues in the body. The true Duffy null phenotype is the result of being homozygous for the FY allele found in populations besides African Americans. This is a very rare silent allele that produces no gene products. This allele is the result of point mutations that encode for premature stop codons in either the Duffy A or Duffy B alleles. No Duffy antigen will be present on the red cells and other tissues in the body. These individuals can make anti-Duffy A, Duffy B, and Duffy 3. Duffy X, also called Duffy mod, is essentially just a weakened form of Duffy B. Duffy X is caused by a single nucleotide polymorphism in the Duffy B allele that results in weakened Duffy B antigen expression. This mutation is mostly found in Caucasians. Individuals with this mutation may type as Duffy B negative with some commercial anti-Duffy B reagents. It is important to note that Duffy X is a phenotype and not an antigen, and there is no such thing as anti-Duffy X. The Duffy protein is an integral multipass glycoprotein. It passes through the red cell membrane 7 times. Duffy A, Duffy B, and Duffy 6 are located on the extracellular amino terminus of the glycoprotein. Duffy 3 and Duffy 5 are located on the third extracellular loop. The Duffy glycoprotein, called the Duffy antigen receptor for chemokines, functions as a red cell receptor that binds and sequesters chemokines released during inflammation. It is thought to act as a scavenger, reducing excessive amounts of toxic chemokines in circulation. Animal studies have suggested that the function of the Duffy gene is not physiologically important because mice that lack the mouse homologue of the Duffy gene were not more susceptible to infection than mice that express the gene. Duffy A and Duffy B negative red cells will not bind chemokines. The Duffy antigens are well developed at birth and found on red blood cells and other tissues, such as the brain, colon, endothelium, lung, spleen, thyroid, thymus, and kidney tissues. Duffy antigens are known to deteriorate in storage, so it is important that when you are testing with out-of-date cells to confirm the antigen presence when ruling in or ruling out antibodies. Duffy A, Duffy B, and Duffy 6 antigens are destroyed by enzymes such as thiosin, papain, and bromelain. All Duffy antigens are resistant to DTT and acid. Duffy 3 and Duffy 5 are high incidence antigens that are not destroyed by enzymes. If a person is either Duffy A and/or Duffy B positive, they will also be positive for Duffy 3 and Duffy 5. However, Duffy 5 will not be present on Rh null cells even if they are Duffy A or Duffy B positive. Duffy 3 and Duffy 5 antigen expression will be weakened on red cells with the Duffy X phenotype. The Duffy 6 antigen is a high incidence antigen that will be present on L cells that are either Duffy A or Duffy B positive. The Duffy 6 antigen has only been defined by a marine monoclonal anti-Duffy 6. To date, no human anti-Duffy 6 has been found. Anti-Duffy 6 has a specificity similar to anti-Duffy 3. The Duffy 6 antigen will be destroyed by enzymes, unlike the Duffy 5 and Duffy 3 antigens. In this table, you can see the phenotype frequencies for the Caucasian, African American, and Japanese or Asian ethnic groups. It is important to note that African Americans will be much more likely to be Duffy A and Duffy B negative, and that Japanese or Asians will be much more likely to be Duffy A positive. You should be familiar with the phenotype frequencies across these three ethnic groups, as they often come up in exam questions. The Duffy antigens are receptors for the malaria parasites Plasmodium vivax and Plasmodium knowlesi, which invade at the Duffy A and or Duffy B antigen sites, causing malaria. Duffy A negative and Duffy B negative individuals are immune to infection from Plasmodium vivax. In the malaria epidemic regions of Africa, the GATA1 mutation in Africans emerged as an evolutionary protection. Duffy antibodies are clinically significant IgG antibodies comprised mostly of the IgG1 subgroup. They are best reactive by the anti-human globulin test. Duffy antibodies are known to show dosage. This means they react stronger with homozygous versus heterozygous cells. Duffy antibodies cause delayed hemolytic transfusion reactions and can cause hemolytic disease of the fetus and newborn. Anti-Duffy A is approximately 20 times more common than anti-Duffy B because Duffy B is a much worse immunogen than Duffy A. Anti-Duffy 3 is a high-incidence antibody made by immunized Duffy A and Duffy B negative individuals. Anti-Duffy 3 will react with all cells except Duffy A and Duffy B negative cells and is not sensitive to enzymes. An interesting fact is that anti-Duffy 3 made by Caucasians will react with cord cells, while anti-Duffy B made by African Americans will not react with cord cells. Anti-Duffy 5 is a high-incidence antibody with a specificity similar to anti-Duffy 3, except that anti-Duffy 5 will not react with Rh null cells regardless of their Duffy antigen typing. The Duffy gene produces the Duffy antigen receptor for chemokines. Duffy antigens are expressed on red cells and other tissues in the body. The Duffy A, Duffy B negative phenotype is resistant to infection from the malaria parasite Plasmodium vivax. Duffy A and Duffy B negative individuals are more common in African Americans due to the GATA1 mutation found in this population. Duffy antibodies are clinically significant and comprised mostly of IgG1. Duffy antibodies show dosage. Duffy A, Duffy B, and Duffy 6 antigens are sensitive to enzymes, and all antigens are resistant to DTT and acid.